Hello everyone. Scholar Labs is an innovative virtual platform that brings science experiments to life through 3D augmented reality. Our simulations, designed by expert educators, enhance the learning experience and are accessible on all devices without needing special equipment. Today, I will be taking you through the simulation Nutrition in Plants. In this simulation, you will be able to understand the different modes of nutrition in plants. Look for the I button on your screen to have the overview of the simulation and for the step-by-step instructions to navigate through it without any hassle. You already know that food is essential for all living organisms. The process of obtaining food and utilizing it to grow, stay healthy, and repair any damaged body part is known as nutrition. Did you ever wonder as to how plants get their nutrition? What do they eat to grow? Run the simulation to figure it out for yourself. Let's understand the different modes of nutrition in plants one by one. The first mode of nutrition in plants is autotrophic nutrition. Auto means self and troph means nourishment. So, autotrophic nutrition is the mode of nutrition in which the organisms make their own food. Therefore, plants are called autotrophs as they are able to produce their own food through a process called photosynthesis. So this process requires chlorophyll, which is a green colored pigment, sunlight, carbon dioxide, and water. And usually it takes place in the leaves of the plants. Let us figure out the importance of each of these factors in photosynthesis. For this, click on autotrophic and then click on water. Click on the watering can to start the experiment. In this experiment, one pot is watered regularly, that is plant A, and the other plant, that is plant B, is not watered at all. Scholar Virtual Lab often incorporates interactive elements and animations that can enhance understanding in ways that might not be possible in a traditional lab setting. You will observe that in absence of water, the leaves and stem of the plant become limp and droopy. Leaves turn yellow or brown as the plant starts to dry out. Plants shed their leaves and photosynthesis does not take place, ultimately leading to death of the plant. Click on this button to go back to the main screen. Now let's observe what happens if the plant does not get sunlight at all. Click on this watering can to start the experiment. In this experiment, both the plants are watered regularly, but plant A is kept under sunlight and plant B is kept in shade. Scholar Virtual Lab is a valuable tool in teaching plant nutrition, particularly in scenarios where traditional lab experiences may be challenging to implement. You will observe that after few days, the plant receiving sunlight shows proper growth. On the other hand, even if we water the plants regularly but without adequate sunlight, leaves lose their color and the plant eventually dies off as they are unable to perform photosynthesis. Let's get back to the main screen by clicking on this button. Now let's investigate the need of carbon dioxide in photosynthesis through Mole's half-leaf experiment. Click on the arrow button on the bottom left corner of the screen to expand the menu and view additional features. Click on this button to hide or show the labels. To zoom in or out, use this mouse slider at the bottom left corner of your screen. For this experiment, you need a destarched potted plant which is placed in complete darkness for 2 days. Now, click on potassium hydroxide bottle to insert about half of the portion of a leaf of this destarched plant into the bottle. Potassium hydroxide absorbs all the carbon dioxide from the leaf. Then place the entire arrangement in sunlight for about 8 hours. Experiments which involve handling of chemicals or working with delicate equipment can pose safety risks. Scholar Virtual Lab allows students to experiment and learn without the risk of injury or exposure to hazardous substances. Again, click on the bottle to remove the apparatus. Click on the leaf exposed to potassium hydroxide to keep it in the Petri dish. Now, click on the leaf which was not exposed to potassium hydroxide and put it in the Petri dish. Click on the Petri dish to keep it on the tripod stand. And then click on the burner to place it under the Petri dish. Now, pour some hot alcohol in the Petri dish by clicking on the beaker. Switch on the burner by clicking on the knob. Alcohol decolorizes the leaves by dissolving chlorophyll present in them. Click on the beaker full of water to rinse the leaves and again put them in the Petri dish. Now let's test the leaves for the presence of starch. Click on the dropper to put a few drops of iodine on the leaves. Click on the leaves to observe the effect of carbon dioxide on photosynthesis. You can see here that in the first leaf, the portion of the leaf which was present inside the bottle of potassium hydroxide shows negative starch test. That is, it does not change color because in the absence of carbon dioxide, the plants cannot produce starch. Even though this part was receiving enough sunlight and water, still photosynthesis did not occur because it was devoid of carbon dioxide as the latter was absorbed by potassium hydroxide. In contrary to this, the part of the leaf present outside the bottle changed its color to blue-black, indicating the presence of starch. Similarly, leaf B also showed positive starch test because all the essential requirements for photosynthesis, that is light, chlorophyll, water and carbon dioxide, were readily available to it. Go back to the main screen by clicking here. Let's perform another experiment to show the presence of chlorophyll in leaves and its effect on photosynthesis. For this, you require a de-starched potted plant, a tripod stand, Petri dish, hot alcohol, and a timer. Keeping the plant away from sunlight for 2-3 days halts the process of photosynthesis and de-starch the plant. Click on the Petri dish to place it near the plant so that the leaf can be kept in the Petri dish. Now, pour hot alcohol in the Petri dish by clicking on the beaker to immerse the leaf in it. Open the window of the room so that the plant gets enough light for photosynthesis. Have a look at the timer. The typical wait time for this experiment is approximately 8 hours. But with Scholar Virtual Lab, you can perform such time-consuming experiments with ease in just few minutes with few mouse clicks. After 8 hours, you will observe that the leaf slowly loses its green color, which goes into the alcohol. Again, click on the Petri dish to see the results. The first leaf is green in color, showing the presence of chlorophyll. If you perform the iodine test on this leaf, it will turn blue-black, depicting the presence of starch. However, the second leaf, which was decolorized in the experiment, has turned white, showing the absence of chlorophyll. If this leaf is subjected to iodine test, it will not change color and show the absence of starch. Click here to go back to the main screen. Some plants do not contain chlorophyll and hence cannot synthesize food. Like humans and animals, such plants depend on food produced by other plants. This type of nutrition in plants is referred to as heterotrophic mode of nutrition. There are few plants which can trap insects and digest them. Such insect-eating plants are called insectivorous plants. Click on the object at the top right corner of the screen to release insects and see how insectivorous plants like pitcher plant trap insects and feed on them. Pitcher plant has modified leaves which look like a pitcher or jug. The apex of the leaf forms a lid which can open and close the mouth of the pitcher. It also has downward facing microscopic hair. When an insect lands on the pitcher, the lid closes and the insect gets trapped. These plants digest the insects by secreting digestive juices and absorb the nutrients from them. These plants usually grow on the soil that lacks minerals. Now let's go back to the main screen. Another mode of nutrition in plants is saprotrophic mode of nutrition, in which the plant takes nutrients from dead and decaying matter. For detailed information on this topic, kindly refer to the simulation Fungi in Microorganisms. Thanks for watching.